Okay, so this is the second part to the T cell for this unit, and so this chapter looks at T cell activation and then some of the differentiation that happens to it. So at this point, the T cell is already developed in the thymus, and it's left the thymus as an emergent T cell. So it would be a single positive T cell, and now it's going to the secondary lymphoid tissue. And so for T cell activation, we need to have 3 signals happen. There is the 2-signal hypothesis, and then what happens is there's a third one. Okay, so what we need to have is the first signal comes from antigen presentation. So antigen is going to be presented on an MHC to the T cell through its T cell receptor. And so we need to have those interactions between the MHC and the T cell receptor. So that gives us our first signal. And then we need to have a second signal in order for activation to happen, to have an effector function. And so this comes from our co-stimulatory molecules, which will then allow for signal transduction and activation of cell signaling pathways so that we would actually get activation and differentiation of the T cell, so they'd have their end effector forms, so that you would have those CD8 T cells that would be able to kill. So they would be cytotoxic. And then you would have CD4 T cells that could differentiate into different subtypes, like your helper cells, like your memory cells. Okay. So this figure just gives you a nice overview of where this is happening, what's happening during this process. So you'll notice in the lower part of the figure, the idea that this is happening in the lymph nodes. So that purple structure there you see are the lymph nodes. That we would have antigens that are present, in circulation, but then we would get interactions happening in the lymph node that would allow for the activation and then differentiation of our T cells. So we can see in the upper panel, starting at the left, we have antigen recognition. So again, we have an antigen presenting cell that's going to be presenting antigen to the T cell. And then you can see how there's interactions between the MHC that's on the APC, on the antigen presenting cell, and then we also have the T cell receptor forming a complex. You will notice that there's also other the co-stimulatory molecules that are involved there. So you would have your CD3, you would also have CD4 and CD8, depending upon what T cell you have. This then leads to activation so that we then get cytokine production. And that's really your third signal, is that cytokines are then that are produced by the cell because we have that triggering, if you will, of your antigen recognition from the T cell receptor and the co-stimulatory stimulation that causes the production of the cytokines. And because that cell has those receptors, they can sense those cytokines, so that causes additional changes, further activation of that cell. So what's going to happen is that you're going to get proliferation of the cell. So what we're seeing is clonal expansion in a normal cell, we just call it proliferation. But proliferation, clonal expansion, where we're making copies of it. Again, we've already locked in that T cell receptor, so all of those cells would have the same T cell receptor. And you're also getting other cytokines that are also feeding in because your antigen presenting cell, helper cells, regulatory cells, all can also produce other cytokines that will feed in and help with clonal expansion and differentiation of these cells. Okay. Now looking at our CD4 positive that are shown in the blue, those cells can differentiate so that we can get a memory population, but then we can also have a helper population. So helper cells are abbreviated TH. Memory, they don't usually get much of an abbreviation. You might do MEM, and then T, or T and then MEM. So sometimes the letters put before the T and sometimes the letters put after the T. So just be aware of that. Okay. So these cells, these helper cells can go on to help to activate macrophages, B cells, and other cells. When we look at the CD4 positive T cells, the helper cells, they're really important. Just like the regulatory T cells are really important in modulating the immune system, usually in a helping, activating, let's go do something way, where regulatory cells are in more of a quelling, a quieting way. Okay, so the two of them you can kind of think of them as a yin and yang. Memory T cells also play an important role because if we've seen an antigen once, we'll probably see it again. And so we don't want to have to go back through this process all over again. The memory allows us to kind of jumpstart it. I would say that memory is looked more at in B cells, but there is a growing area of research looking at memory in the T cell population. Okay. Now, looking at our ruddy orange or rusty colored cells that are CD8 T cells, when those undergo clonal expansion and they differentiate, they are going to differentiate into an effector function, which would be the cytotoxic T cell, or they can differentiate into a memory population. So you would have memory CD8 positive T cells also, along with our memory CD4 T cells. Okay, that memory cell can differentiate into the cytotoxic T cell. Just be aware of abbreviations for cytotoxic T cells. You can have TC or you can have TCL, and there's TCYTO. So cyto being a subscript. Okay. So for our cytotoxic T cells, their effector function would be that they're going to go into peripheral tissues and they're going to, engage those target cells. They can produce porphyrins that will cause holes on the surface. So porphyrins are very much are similar to the membrane attack complex, where they embed these proteins in, and then we get this pore formed, so the cell will actually lyse. But it can also be really important for macrophage activation also. So just looking a little bit more at the signaling, so this figure nicely looks at the different, the three signals that are needed. And so again, the first signal is going to come from that T cell receptor signaling, because it's going to be interacting with the MHC from the antigen presenting cell. Now keep in mind, just the interactions between those receptors aren't sufficient. We also need to have that antigen, that processed peptide in there binding between the two. Okay, I just wanted to point that out. For your second signal, again, this is going to be co-stimulatory molecules that are going to bind to each other. You're going to have co-stimulatory molecules on the APC, antigen presenting side, and also on the T cell side. And that when you have those two signals, that causes gene expression change. And so we also are having cytokine signaling from our other cells, but then we're also having autocrine expression of cytokines that feed back to the cell itself. So do be aware, for cytokine signaling, there are paracrine cytokine signaling produced from another cell and feeds to our T cell. And we also have autocrine, and that is where autocrine, the cell, is going to produce the cytokine that it senses, so by itself. Okay, so those cytokine signaling is really important too. And so between all of this, the T cell is taking in all this information, how strong each of those are, to then indicate what should the response be. So what kind of cell am I going to become? I'm going to get activated, but what does it mean? Okay, what does it mean for me? So looking at, when we have these successful interactions, this would be called an immunological synapse. Okay, so just like we would have like a neuromuscular synapse, where we're going to have interactions between a neuron and a muscle, the immunological synapse is those interactions between the T cell and the antigen presenting cell, the APC. And the signaling molecules that are involved in that process. So we can see here on this figure how on our panel B, on the left-hand side, we have our CD4 T cell, and you can see the different surface molecules that we have. So we have like CD2, CD4, CD3 complex. We have our T cell receptor. We have LAF1, but then on the antigen presenting side, we also have molecules that are going to interact with those. We'll look more at those molecules in a moment, but I just wanted to point some of those out. Now, when we look at our CD8 T cell, which is on the right side of that lower panel, again, we can see that we have these surface molecules that are present on them, and we also have those molecules that are present on our antigen presenting cell. Okay. One of the things you'll notice is that for the CD4 T cells, that it's MHC class 2 that it's interacting with. CD8 T cells can interact with the MHC class 1. So this would mean that, if you have a T cell receptor that interacts with class 2 better than class 1, that, that would have pushed you back in development towards having a CD4 expression, right? Where if your receptor was better at binding MHC class 1, then that would have given you a CD8 expression. So I just want to kind of link that with our last chapter. Okay. So with this complex, looking at this complex, we're looking kind of at a CD4 positive T cell and looking at those structures in that shaded box, but this easily could be for a CD8 cell if we switched out the CD4 and the CD8. But we would have all of these intercellular signaling molecules that would be involved, and so once we had our T cell receptor and then our co-stimulatory signaling occurring, that would cause activation of these different signaling molecules for signal transduction, right? And so this could be phosphorylation of these different factors. So, through cell bio, you would have talked about some of these. So this should be a little bit of a refresher of some of that. So you can have protein kinase C activation, and that leads to activation of NF-kappa B, but we can also have MAP kinase cascade. So again, the end result would end up being gene activation. Many of these genes that are going to lead to activation would end up being related to proliferation, related to cytokine production, and then effector function. So I want to talk a little bit more about signal 2. So again, signal 2 are the co-stimulatory signals that allow for T cells to become activated and then signal clonal expansion or proliferation. Now, there can be positive co-stimulatory receptors, but there can also be negative co-stimulatory receptors. So the positive ones are CD28 and ICOS, and then the negative co-stimulatory ones are CTLA4, PD1, BTLA. And this table from the textbook looks at these both stimulatory and inhibitory receptors and what their activity is. And consensus is that if it's stimulatory, it's going to end up activating, leading to differentiation of our T cell. With inhibitory, then it's going to negatively regulate the immune response so that the T cell doesn't become activated. It can also cause the T regulatory cells to be activated. You'll also notice that with the co-stimulatory receptors and the co-inhibitory receptors, that they have shared ligands. So keep in mind that the ligands are going to be found on the antigen presenting cell. And so this allows for, like CD80 to be able to bind either to CD28 or to CTLA4. So, depending upon which one it binds, would end up giving you a different response, whether you get activation or inhibition of that. So we can also get energy, so clonal energy, if we don't get that second signal. So again, signal 1 is not the end-all be-all. You need to have signal 2 and really signal 3 also in order to have effective response. So this helps to provide tolerance, especially in the periphery, so that we don't have, hyperactivation of a response, especially an autoreactive one. So if only signal 1 is received, the cell would be non-responsive. And so we can see that in panel B there, where the cell doesn't have that co-stimulatory response. And so it would not actually be able to respond, because as you can see here, this is not an antigen presenting cell. This is our cell presenting cell. So we wouldn't want it to respond. Okay. Now, we can also get the energic response because we could have an inhibitory. So you can see on panel C how when we look at the T cell side, we see CTLA4 and we see PD1, and so those are inhibitory, and so by being inhibitory, that leads to an energic response. Now when we have stimulatory signals, co-stimulatory molecules present like CD28, that leads to an activated, so you can see that in panel A. And then again, the third signal is the cytokine production. And so if we do not have the cytokine production, we're not going to get as robust response from our T cell. And so IL-2 is a well-known autocrine type cytokine, where the T cell will actually produce it, and then it also has a receptor. So remember, it had to have an IL-2 receptor in order to exit. And so having that receptor allows it to be able to sense it. Okay. We can also have polarizing cytokines that that send the cytokine down different subsets of developmental paths. Okay. So just keep in mind that we can have the own cytokines being produced, but we can also have these paracrine cytokines, and then we can have cytokine, and it's not that, cytokine's there, we get this response. It depends upon what cytokine is present that ends up giving us our different subset that arises. So antigen presenting cells have co-stimulatory properties, so they are going to be able to provide those signals because they have the surface ligands in order to activate. And so you can see here how we have dendritic cells, macrophages, and B lymphocytes. They're going to act as antigen presenting cells, and you can see how on their surfaces they have the MHC to give that first signal, but they also have those co-stimulatory ligands that are going to bind to the receptors. And so that when they become activated, they increase production of those. And so we can use those markers to indicate whether those antigen presenting cells like dendritic cells, macrophages, and B cells are activated. And once activated, they can help in presenting and activation of our T cells. So there are some super antigens. These are kind of special T cell activators. So they can actually short circuit co-stimulatory signals. So they can actually kind of bind in place and stabilize the T cell MHC interaction so that you don't require the costimulatory signal to like you normally would. So some of these real-world applications of super antigens would be some of the foodborne pathogens, so food poisoning. So we have different staphylococcus, usually aureus, enterotoxins that can be produced, and so that these can actually bind to the beta chain and help to stabilize the interaction between the T cell receptor and the macrophage. Also, the toxin that's produced in toxic shock syndrome. There's also scalding skin syndrome, where the skin basically just kind of separates, and so this is actually due to a super antigen that's produced by Streptococcus. And then we also have mycoplasma supernatant, and this actually has like an arthritis type syndrome. But we can also have Streptococcus that's also can produce exotoxins, and so that can lead to rheumatic fever and shock, and so that acts as a super toxin. So you get this hyperactivation of the immune system with those. And the reason why you're getting this hyperactivation of the immune system is because normally you would have those co-stimulatory signal requirement, and that would help to regulate the response and the activation of the T cell, but that's been bypassed by these super antigens. Okay. So one of the things we want to keep in mind is that when we activate our T cell here, again, you can see our first signal and our second signal, and then we're getting our cytokine production, and so that's activating, so it becomes it proliferates. One of the things that happens is that over time, as it terminally differentiates, its ability to self-renew is decreased. So the ability to make copies of itself, again, is going to decrease as it gets terminally differentiated. So when we look at memory cells, they still have that self-renewing capability, so that they would be able to proliferate, make copies, undergo clonal expansion. Okay, but again, terminally differentiated cells, when they're effector cells, are not going to be able to undergo that self-renewal at the same rate, at the same number that we would see with those more stem cell-like memory cells. So when we look at T-cell differentiation, we have a number of different subsets that can arise, and it depends upon cytokines that are present in that third signal to what T cell we're going to end up having, what's subset. So keep in mind that the APCs may be binding PAMPs or MAMPs or DAMPs on their pattern recognition receptors, PRRs, and so that's going to induce cytokine secretion from them. And so depending upon what pattern recognition receptors are being engaged, activated, that's going to give different cytokine production, which would, again, give you a different T cell subset. And so when we look at viruses and the pattern recognition receptors they activate, that leads to an IL-12 production, which shifts to a Th1 subset. And we'll talk more about these subsets in a little bit. Where if we have parasitic worms present, this actually produces an IL-4 production, and that induces a Th2 subset. And so again, there's a lot of factors contributing to what cell type subset you end up getting with your cells, right? Some of that information is coming before the T cell even arrived on scene. It's from what the APC saw, because based on what it saw, because of the ligands that bound to its receptors, it needs this type of cell to fight this type of infection. Okay, so again, here we see our antigen presenting cell. It's got the pattern recognition receptors, and so it's going to recognize those PAMPs, and that's going to cause a certain cytokine production that's going to then turn on effector cytokines that are going to feed back into the different production of the different subsets. So looking at the different subsets, there is this table, and there's also a figure in the textbook that really nicely summarizes up some of the subsets for the T helper subset. So again, keep in mind that these are CD4 positive T cells. And on this table, and also on that figure that we'll show in a moment, you can see what cytokines are being produced that lead to that subset development. You can see what genes are being regulated, so what genes are being turned on in order to make that subset. Effector cytokines, what cytokines are being produced by that subset. And then lastly, what is the function? So what are they doing? And you can see each one has a slightly different function. And so again, depending upon what the response is to, we would want a certain subset to be present, and so we want that kind of response to it. Okay, so not that this is a bad table, but I just like figures, I like visuals. So again, you get this idea that we have these different T cells, Th cells, T helper cells. Our major ones, and I say major only because these are the ones that have been looked at more or for a longer time or were identified longer ago, would be the Th1, which is in our teal color, and then Th2, which is in that blue color. So looking at these ones, so Th1, you'll remember these are initiated with like a viral infection, and so they're going to respond to IL-12. So you can see IL-12 is our polarizing. We also have interferon gamma and IL-18. So it's not just one cytokine causing this T cell to differentiate into a Th1. But then we get production of interferon gamma, so it kind of feeds into the interferon gamma that's already there, and also TNF. So this is going to help with intracellular pathogens. So that works great because that's what viruses are. It also helps to activate macrophages. And so really important when we have tissue inflammation, and a lot of times when we have a viral infection, we're going to have tissue inflammation because the tissue that isn't virally infected is being damaged. Okay. Now, when we look at our Th2 cell, it's going to be IL-4. Again, keep in mind these are the ones where the APC, the antigen-presenting cell, saw like a parasitic worm, and so we're going to get production of IL-4. That cell senses that causes GAT3 production, and that's going to lead to additional IL-4 production, IL-5 production, and IL-13 production. Now, IL-13 production, I would say, like, that's kind of important. You know, we'll definitely have to, you know, we're getting closer to the end of the semester than not, but there's a couple like journal club papers that are really great, and one of them looks at IL-13 production, and what's nice is it's with parasitic worms. So this actually activates those cells that are needed to fight a parasitic worm infection, like xenophils. So xenophils are able to produce paralyzing agents. So they produce chemicals that will paralyze the worms, so they actually disengage. Also important for allergies. So again, I would say with the Th1 and Th2, again, those are the ones I've been studied more so. They exist back when I took immunology 10 plus, way more than 10 years ago. Probably 20 plus years ago. But, you know, these other ones are just as important. T regulatory cells, they're going to use the IL-2 production in TGF beta production, and they induce FOXP3, and then they're going to produce IL-10 is the big one that gets looked at for those. So IL-10 production, and so they're going to help to suppress the immune function. These can actually be really important in tumors, so they actually lead to an anti-tumor response, and so Tregs are being looked at in cancer, because if we can stop those Tregs from telling the immune system not to respond, then maybe we could get a response to them. Tregs are also really important in autoimmunity, but for the other reason that Tregs are found in lower numbers in autoimmunity. And so the issue is, well, how can we increase the amount of T regulatory cells? And some of that goes to parasitic worms. We have these, like, what's the role? But there's a lot of overlap between these. And so we know that with parasitic worms, they actually induce a Treg population. And so individuals who aren't exposed to parasitic worms have lower levels of T regulatory cells, which then potentially leads to autoimmunity. Okay, so one of the mechanisms that are being looked at around the hygiene hypothesis. Okay, so those are the three that I'm going to focus on. So obviously, there's more than three on the figure. You can always pick your favorite one. Which one is related to maybe a disease that you're interested in. And the reality is that this is not a complete list. There are additional subsets that are being identified. And if you remember back to our first journal club paper, now that we can do individual cell analysis of, immune cells that are in circulation, this is going to get even more complex, because the reality is that once it becomes kind of standardized that we would be using a single cell analysis, we'll end up seeing that there's a lot of additional subsets that are involved. So again, I would really love it if everybody knows the Th1, Th2, and Treg. We could add in some additional ones. But again, I think if everybody knows those, I'm super happy in definitely knowing what are the polarizing cytokines, what are the cytokines that are inducing them to become that subtype, and what cytokines are they producing. So as I mentioned, we have the type Th1 and Th2, and sometimes those are referred to as a type 1 response versus a type 2 response because of those different cells that are induced. And as I mentioned, with type 1 response, these are usually intercellular pathogens, and whether that's a virus or whether that's some kind of bacteria that's intercellular, we then need to have a cell-mediated response to that so that we can clear those cells out. And so the Th1 is that helper cell that's going to help to lead to the activation of our CD8 T cells so that they can go off and they can help fight and clear out these infected cells. Okay, so again, we would have that virus inside, so we would, or it's being presented to the dendritic cell, to the APC, and so it's going to present some of those fragments to that T cell on the MHC, giving us our first signal, and then we're going to have co-stimulatory signaling. We're going to get production of cytokines, so we can get our signal 3, and then all that information is being processed between those signals so that we end up getting STAT4 activation to get T beta activation that leads to interferon gamma production, which gives us the Th1. Okay. Now looking at the type 2 response, again, this is usually extracellular parasites, so like worms. So you've got your dendritic cell that's come in and sense the worm being there from its pattern recognition receptor. And so now it shares some of that antigen with the T cell. So we get our first signal, we get co-stimulatory signal for second signal, and we get cytokine production like IL-4. So again, all of this information comes and gets processed in. So we get STAT6 activation that leads to GAT3 activation, and then now we have a Th2 profile, okay, so a type 2 response. So we would get production of cytokines like IL-4, which is already being produced, but further producing that so we can activate additional cells, but also IL-5 and IL-13. IL-13 would feed back and lead to a xenophil activation. It also leads to increased mucus production, so we get better antibody interactions with the parasitic worms. So our T helper cells can also help other cells, and so looking at panel A, we're seeing where our T cell is helping B cells. So this is a T cell, the follicular T cell, and so it's known to help B cells, and so it has antigen that's derived from like a virus. So the B cell's showing it and sharing it, and it's okay, should I respond, should I not? So the T cell can become activated, but at the same time the B cell can become activated also. And so that you can see how the B cell becomes activated, and so it's going to undergo isotype switching, so that it's going to become a mature B cell, a plasma cell, and then be able to secrete antibodies that are against the virus that can neutralize it. Okay, so this would not have happened if that T cell, that follicular T cell, hadn't helped that B cell to become activated and undergo this isotype switching and affinity maturation with its B cell receptor. Okay. We can also have T cells helping other T cells, and so you can see here how we have our Th1 cell, and so it's interacting with the antigen presenting cell, but it also can help to stimulate the CD8 T cell. So you can see how that CD8 T cell is interacting also with that APC, and so we have presentation of these antigens to both of these cell types, but then we have stabilization between the CD40 ligand, and so that's going to help that CD8 T cell become activated. We also have cytokines that are being produced, and so that would also assist in the activation so that the CD8 T cells will proliferate. Some of them can go off to the tissue. We can also have memory being produced also. And as I mentioned, that CD8 T cell, the cytotoxic ones, can produce these porphyrins, and they can get incorporated into that target cell, so that the cell will end up dying. And whether that is a virally infected cell that's self, or whether that's a pathogen. So we want to keep in mind with T cell differentiation, we're talking about these like linear, like, oh, if you're this kind, you have to do this, and then you have to do this. But the reality is there's fluidity, and so that just because you're a certain subtype doesn't disallow you from producing a certain cytokine. So there are some Th2 cells that actually can produce interferon gamma when they get exposed to IL-12. There's also young Th1 cells that will produce IL-4 when they're in these Th2 conditions. So again, we don't want to think of these as these like kind of locked, like this is the response you have to have, and it can't be the cell if it's not. Now, in the future, we might actually identify these as different subsets. So who knows? Time will tell. So helper T cells play a critical role in a number of different human health issues and diseases. So probably the one most people know most about is HIV. So HIV progressions into AIDS is where our CD4 T cells are going to be targeted by the virus and destroyed. What happens is there's actually a shift from the Th1 to a Th2 response over time, and so this gives the virus an advantage. Okay. Leprosy is also responsible or due to a Th1 response. So there tends to be a high circulation of IL-2, interferon gamma, lymphotoxin A. Okay. We also know that with Th17 cells, those are really important in controlling infections with extracellular bacteria and funguses. We also, with viruses can augment the response by the immune system, right? So to evade it. And so we can see that some viruses will actually cause the production of IL-10. And you'll remember IL-10 is one of those cytokines that normally a T regulatory cell would actually produce. And so this actually quiets the immune system, so it will reduce that that Th1 response that we would normally see. So just looking at T cell memory, again, I always feel like B cell memory is looked at more so, but T cell memory is really important. So there's different surface markers that can differentiate our different T cells. And so CD44 is one of those, and we talked about that as an adherence marker. We also have CD62L. So this is another adhesion molecule that actually homes it, so it goes back into the secondary lymphoid organ. So keep in mind that these T cells can go out into circulation, but they're going to come back to like a lymph node. They go out and they come back. Same thing, we have a CCR7, which is that cytokine receptor. So again, allows it to home back to the secondary lymphoid organs after it's had a chance to go to the peripheral tissues. Okay, and then we have CD69. So this is a C-type lectin, and it's going to actually help prevent the immune system from leaving the tissue. So it actually will like stay in that location. So when we look at some of these different markers, you can see how the expression is different on the different T cell populations. So when we have a naive T cell, we are going to see our chemokine receptor upregulated, but when they're in the effector phase, they're not going to have the same regulation of that. And then looking at our different memory populations where we have central memory, we also have effector memory, and we also have resident memory. So resident memory is going to stay in that tissue. You can see how those surface markers are changed, and so you could use that as a way to characterize what population you're looking at. So for our T cell memory, we have different subsets depending upon their location and commitment to effector function. So our central memory T cells, those are going to reside and travel between secondary lymphoid tissue. So again, from one lymph node to another. So these are going to be longer lived and divide more time than our effector memory. So they're kind of more stem cell like. So they have more of that cell renewal possibility. They do get rapidly reactivated when there is a second antigen exposure, and then they can differentiate into several different subsets depending upon the cytokine environment. For our effector memory cells, these are going to be traveling from tertiary tissues, so sites where we might actually have infections occurring, and so they actually contribute a bit to the first line of defenses because they're going to have a potential rapid response because they're more on scene, if you will. So they would be able to shift back to an effector function, so they can't really do different subsets. They only can do effector function after a secondary exposure. Okay, and then we have these permanent residents of previously infected tissues. So if you've had an infection somewhere, we're going to keep memory there so that they can respond to any kind of reinfection. So maybe we didn't clear out the pathogen all the way. We don't want to wait for something to go back into the lymph node before we activate the memories in central memory. We want to make sure that we're going to activate right there on site. And so we can actually, after we've had infections, we could go into a location, do a biopsy, and we would be able to see CD8 positive resident memory in multiple different tissues. So again, if we look at these memory populations and look at their ability to differentiate, it would be that stem cell-like one in the central that are found in the lymphoid tissues that would have the most potential to become different different subsets and different types of cells. Once we look into the peripheral tissue at the effector memory and also these resident memory, that's where the ability to differentiate is not, they're going to be more differentiated, so they don't have the ability to kind of do the self-renewal as much. Eventually memory all wears out, and so it is, if they don't get utilized, we could have death occurring. So, one of the questions you might be wondering is how and when do memory cells arise? So there's many studies still looking at this and trying to understand the memory population, both for B cells and T cells. It seems that memory cells can arise very early in the immune response. So when the initial activation of the adaptive response occurs within a relatively short time of that, you can have activation. So when we're looking at the immune response, 3 days, we can start to see memory populations occurring. Keep in mind that the first 24 to 48 hours would be innate response, and so we're talking about adaptive cells being activated at day 1, day 2, and so this is relatively shortly after that. Usually your central memory cells are going to arise prior to your effector memory, and it's not that that's where your effector memory are coming from. Effector memory can actually be derived from differentiated effector cells. And just keep in mind that it's not a one here, one there, one there, that there's really an asymmetric division of activated T cells. And then lastly, with self-renewal memory cells populations, they may be generated during T cell activation so that we would have that population present. So what cells induce memory cells? So it seems like T cell help is required for that to happen, and IL-7, IL-15, and the Wnt pathway may play a critical role for the stem cell memory cells, and it seems like IL-2 is important for generating the effector memory cells. So do memory cells reflect the heterogeneity of the effector cells within the primary response? I don't know if I said that. Do memory cells reflect the heterogeneity of effector cells in the primary response? And that's unclear. So it seems like the memory cells that are induced are diverse. And so whether the original response actually provides, what cues that original response actually provides to generate the memory population really needs to be further explored. So what's the differences between the CD4 and the CD8 memory T cells? Generally, there's more CD8 memory cells present than CD4 memory cells, but they both proliferate during the response, so they are present. And some of that might be due to the differences in lifespan between the two memory populations. So CD8 memory tends to be longer lived, and so that's why it may appear that there's more of them. Okay. How many memory cells are maintained over the years? Well, that's really unclear. And again, this is where generally the B cell populations looked at more, because we're looking at the antibody response when we're doing like what's the memory population, like in a clinical trial and such. Okay, so it becomes unclear how long they do persist when there's an absence of antigen. So definitely an area that needs further research to understand how many cell divisions they can undergo, and then what cytokines are important to kind of bring them out so that they go out of the memory stage and start dividing. It does look that like IL-7 and IL-15 are important in allowing for proliferation to occur. So in summary, with T cell activation, there's many aspects that are well defined and understood, but there's definitely, when we're looking at the different subsets, there's further work that needs to be done to further characterize and really understand what what's occurring, and how could we actually go and push to different subsets and such, and how can we utilize that in different disease models. And then, to further that, those memory populations, that's definitely an area that needs further research, needs further elucidation of how do we get these different memory populations and what roles do they play in our general health, but also in different states of disease. So this concludes the T cell chapters that we'll be covering for this unit. We will be talking about B cells in our next 2 audio lectures, so B cell development and also activation. So definitely we'll talk about T cells, but again, the focus won't be on the T cell, it will be on the B cell. And as always, if you have questions, please ask.